All right, we'll likely have a few more folks joining us, but we'll go ahead and get started. So good afternoon. My name is Lisa Gerloff, and I am the Executive Coordinator for the Rocky Mountains Cooperative Ecosystem Studies Unit, or CESU, and I welcome you to our 2022 online seminar series. Our seminar theme this year is celebrating the scientific and scholarship contributions of our Rocky Mountain CESU Student Award recipients. We started our Student Award program in 2007 as a way to recognize the outstanding work our students were doing on CESU projects. And this has been a really fun theme for us, recognizing the work of our most current award recipients, and then checking in with past recipients on where they are and what they're up to now. So we started the series back in January with our 2021 recipients, Scarlett Engel and Jacob Oram. Scarlett walked us through a culturally appropriate museum redesign for Chapin Mesa and the tribal outreach conducted in order to do that. And Jacob demonstrated how statistics is helping in white bark pine conservation. In February, just a couple weeks ago, Jeremy Sultanis joined us. Jeremy was one of our 2017 award winners, and at the time was working on his PhD at Colorado State University. Jeremy is now a faculty member at CSU with graduate students of his own, some of which are funded through our CESU mechanism. Within impacted mountain systems, Jeremy's talk pointed out why restoration is hard, especially on public lands where there's competing priorities. And using examples from his work and his graduate students' work, while building relationships with managers and working collaboratively is the best way to inform restoration and management of these impacted areas. All of these talks were recorded and are available on our website, and I encourage you to check them out. Today's seminar is being recorded as well and will be added to that list. And I'll put our web link in the chat box for you. As we move into today's seminar, I want to point out that the talk will take about 40 minutes, and then we'll have plenty of time for questions afterwards. And I'll ask you to please use the question and answer box to type in your questions. I'm excited to have our 2016 Rocky Mountain CSU Award recipient with us, Greg Wann. At the time of his award, Greg was working on his doctoral dissertation at Colorado State University. He was nominated by his advisor, Dr. Cameron Aldridge, and Dr. Zach Bowen, who was branch chief at the time at the USGS Fort Collins Science Center, for his contributions to the CESU project, Demography and Vulnerability of Grouse Populations, October 2014 to October 2016. In the nominations, Drs. Aldridge and Bowen noted that Greg's research will directly help managers within the National Park Service and the U.S. Forest Service to better understand population dynamics and potential management and conservation strategies, given climate change may drastically alter habitat quality and connectivity for alpine species. They went on to emphasize, while whitetail ptarmigan are being considered for listing by the U.S. Fish and Wildlife Service under the Endangered Species Act, Greg's science is already helping to inform that listing decision process. Greg serves as an expert scientist on the species and is working directly with the Fish Wildlife Service team responsible for that listing decision. He is recognized as one of the world's leading authorities on whitetail ptarmigan, even though he is a doctoral dissertation. Today, 6 years later, Greg is a research ecologist at the USGS Fort Collins Science Center. He works primarily on conservation issues in the sagebrush and alpine biomes of Western North America. Recent research projects include identifying priority habitats, promoting greater sage grouse persistence, and assessing the population viability of whitetail ptarmigan along Colorado's Front Range. We're happy to have Greg with us today, and I turn it over to him to share more on his research work.
Yeah, hi everybody, and thank you, Lisa, for that introduction. I appreciate it. It's good to have the opportunity to give this seminar today, and thanks to the RM-CESU for the invitation to do this. So today I'm going to be talking about a couple of components of, I guess, kind of the research that I've done over the past 9 or 10 years. And this is going to include some of my graduate research. And then I also picked an example of some work in the sagebrush biome of Western North America to, I guess, just kind of give some idea of the type of work that I've been doing. So this is just an overview of what I plan to talk about. So I had a little bit of difficulty trying to figure out what would be the most appropriate research projects to discuss for this, because I've done some work in other parts of the country as well, including the Southeast with bobwhite and rough grouse and turkeys, some applied projects. But I thought maybe for this audience, it would be best to stick to some examples of work in the Western US, because that's what people might be the most familiar with. So the first thing I'm going to talk about is actually my dissertation work for 1 or 2 of the chapters that went into my dissertation for population ecology of whitetail ptarmigan. And then the second example that I picked was from my postdoc with USGS. I worked for a couple of years out with the WERC group, and that was centered out in Dixon, California. So it's out of that field office. So I'm going to talk a little bit about some sage grouse monitoring programs that we were evaluating for that work. And then I've got a slide or 2 at the very end just to kind of give some information on what I do now and the types of projects that are keeping most of my time occupied. So I just wanted to put this figure on here. This was from a study published in Science in 2019. And this was a study that got quite a bit of attention in terms of news headlines. So it talks about from the 1970s forward how many birds we've lost in North America. So according to the study, we've lost about 3 billion birds since that time. And one of the things I kind of like about this figure is it kind of breaks down by ecoregion the areas that are losing most birds. And if we look at this, we can kind of see that the grasslands, that's sort of the place where most of the losses are happening, but also up here on the Arctic tundra, that's pretty far up this list as well. So the 2 things that I'll be talking about today with ptarmigan and sage grouse both fall into these categories. So I'm going to start off with my dissertation work on whitetail ptarmigan and show you some of the stuff that we've learned about this. So first, a little bit of background on whitetail ptarmigan. This is our smallest species of grouse. It's an alpine endemic, and what I mean by that is it's a species that spends almost its entire life history at or above tree line. It only occurs in North America, so it's very much tied to the alpine ecosystem. So its distribution basically stretches from southern Alaska all the way to northern New Mexico, roughly. There's some question there if the populations have persisted in New Mexico, if if there's been a recent extirpation or not event, but that's the historic distribution here. And on this map here, you can also see these black dots. These are locations where ptarmigan did not historically occur, but translocations put them there. So almost all these translocations took birds from Colorado and moved them to these other areas. So I need to give a little bit of background on how this project started. So the project started in Colorado with a graduate student. His name was Clay Braun, and Clay Braun was interested in studying the effects of hunting on whitetail ptarmigan. So Clay chose 2 study sites that ended up becoming long-term sites where we go back year after year to collect information on ptarmigan. So the site on the left, that's Mount Evans. And that's a site where there was hunting pressure of various degrees over the years. So until basically a ban went into effect in 1995, where hunting was restricted from that area. But prior to that, there was hunting at Mount Evans. The other site was at Trail Ridge. That's a site that's located within Rocky Mountain National Park. And the Trail Ridge population does not have hunting because it is in a park. But it does have some other dynamics going on there. Particularly from around the mid-1970s, elk population started to really increase, and elk will browse willow. So willow is a really important food source for ptarmigan, especially in the winter and the spring. That's pretty much the only thing that they're eating in Colorado during that time. So there's some important dynamics to consider that are sort of differ between these two sites. But the idea, whenever this started, was to use Mount Evans basically as a sort of a treatment site for hunting, and then the Trail Ridge site at Rocky Mountain National Park was a control site. So for my dissertation work, I also collected VHF telemetry data at these sites from 2013 to 2015. And that's in addition to some of the work that we were already doing at these sites over all these years. So we were, so basically there's this continuous history of long-term data collection in the form of spring and summer surveys where mark recapture data are collected at these sites. And that mark recapture data lets us estimate demographic parameters like annual rates of survival and recruitment. So back to the VHF telemetry for my dissertation. So from 2013 to 2015, I also collected VHF telemetry location, which lets us get some additional information that we can't get from mark recapture alone. And that's going to include information that allows us to find females on nests, and then also track females that raise chicks to get estimates of survival, direct estimates of survival on chicks. So we also included another site for my dissertation work. This is at the Mesa-Seco site located in southwestern Colorado in the San Juans. So we did the same thing at this site that we did at Mount Evans and at Trail Ridge with the VHF telemetry data collection. So here's a map of those locations. We've got the Trail Ridge site, that's the one that's farthest to the north. The Mount Evans is in the center there, and Mesa-Seco down to the south. So what's been happening at our long-term study sites over these 50 years? So it's kind of I think pretty clear from these patterns that at our Mount Evans site, from 1966 onwards, we generally had a population trend that was increasing. And then if we look at the Trail Ridge population, we've got kind of the opposite going on here. So around the mid-1970s, we started to see some pretty steep declines in the Trail Ridge population. I don't think I mentioned this in the previous slide. We did have a gap in the data at Trail Ridge. So from roughly 2001 to 2009, we weren't able to go out. We actually didn't collect data during that time period. There was nobody there to collect it because Blake Braun, the guy that kept these sites going, retired from Colorado Parks and Wildlife. But we're still able to produce some estimates in this population model. And that's basically what this is showing right now. It's showing the population estimates of abundance over this time series. So this is a bit more formal. It shows you the estimates of the mean population, the mean annual population growth rates at these sites. So basically this red vertical line right here, this shows a stable population at red. What we can see here is that these posterior distributions, these mean annual growth rates for the Mount Evans population are right of the vertical red line. So that population has been greater than 1, growth has been greater than 1, so it's been increasing. In contrast, if we look at the Trail Ridge populations, we see that for both those distinct periods of data collection, the posterior distributions tend to be left of that red vertical dashed line, meaning that there's been decline. So this is the figure that shows the annual rates of fecundity at Mount Evans on the left and Trail Ridge at the right. So fecundity, what do we mean by that? Well, we mean the average number of chicks per hen that are observed in this summer population. So that's what we're using as our metric of fecundity here. So at both sites, we've seen some trends generally of declining, but that trend has been a little bit stronger at Trail Ridge. So we've seen that the number of chicks per hen has gone down more strongly at the Trail Ridge site. So this decline in fecundity was, to us, really interesting, and it spurred some additional work on that reproductive ecology, that started in 2013 during my dissertation. So we think that we thought that reproductive biology ptarmigan was particularly interesting given that they are in that highly seasonal alpine ecosystem. So females, they can only produce one brood per season, and the renesting rates are really, really low. So if they lose a nest early in the nesting cycle, they're not likely to try to renest to produce another one. So what that really means is that the opportunities for them to breed again if a nest is lost are just very small. And then if you put on top of that the very highly seasonal nature of alpine systems with really, really short growing seasons, you can see that there's like kind of essentially some very, very strong constraints there on reproduction. So one of the first things I was interested to do as a graduate student was just try to look at some long-term trends in breeding, but also see if that was tied at all to weather events. So one of the first things that I actually looked at was relationships between measurements of breeding phenology, so the timing of the breeding events with respect to some temperature-based variables and precipitation-based variables. And we found that there actually is a pretty clear pattern here. So the plot on the left here shows the date of hatch advancing as a function of growing degree days. So that's kind of like a heat measurement metric that's used to it basically provides information on when a threshold above which like plants can grow and insects can begin to reproduce. So what we see there is that as things warm up in the alpine, ptarmigan respond to that by breeding earlier. Now, in contrast, they are ground nesters. So whenever we have snow on the ground, that's going to cover up the access to nest sites. So that tends to actually delay timing of nesting. So what's that done to the populations in terms of trends? So overall, the springs have been warming at our study sites, and as a result, the birds have been nesting earlier. So on average, ptarmigan advance their nesting, their timing of breeding, 1.9 days per decade at Mount Evans and 3.7 days per decade at Trail Ridge. So that kind of brought us to this natural question, which was to ask, so given we're observing changes in reproductive phenology, how does phenology influence production in ptarmigan? And this was particularly interesting to us, excuse me, in light of those trends that we saw in a reproductive fecundity. So in ecology, there are a lot of studies that have documented the importance of synchrony of reproductive events with resource abundance. So this is a conceptual graph by Miller and Rushing, and it basically illustrates how phenological relationships can vary. So in this figure, the dark solid line represents a focal species, and the environmental resources are shown in the dashed lines. So these extra rows below just kind of show that the shape of these distributions can be, can change. So they can either be kind of narrow or wider and stuff. But what happens in a scenario where maybe a focal species becomes out of synchrony with its resources. So if we take a trait, for example, timing of breeding, and we look at the distributions, if they pull apart, you can obviously see that the shaded regions over here on the left are going to become less and less the more these two distributions separate. So that's important because obviously the difference in these timing events, the greater it becomes, the more likely there is to introduce asynchrony. So that's often called the phenological mismatch in the ecological literature. So what's the evidence for this occurring in birds? Well, we really have two types of avian systems where a lot of work has been done on this. And one of those is with migratory passerines, so birds that are basically breeding at summer areas and then making long distance movements to wintering areas. And there are a lot of reasons to think that they might be more affected by phenological mismatch because they're not able to pick up on those direct cues on the in terms of when to breed on the breeding grounds because they have to migrate there from the winter grounds. So they might not be able to time their migratory events to kind of keep up with those resources quite as well. And there is quite a bit of evidence that this signature exists from many passerine studies. So one of the really well-known ones are pied flycatchers that migrate from Europe to South Africa for winter. So they've been declining as a result of this phenological mismatch, which has been observed. Now, in contrast, resident passerines, there are reasons to think that maybe opportunities for phenological mismatch are going to be more limited for this. And the reason for that is that they're going to be basically on the breeding grounds year round, which means they have an opportunity to probably be able to time their breeding events more closely because they can see all the cues about when they need to breed. But there is still some evidence that phenological mismatch can happen in resident species. So there are two really famous studies from Great Tits in the United Kingdom and then in the Netherlands. So the population in the United Kingdom, appears to be keeping pace with changing resources in terms of the phenology of the insects. So they're able to breed or adjust their timing to keep pace with that. But in contrast, in the Netherlands, that's not the case. Apparently there is a phenological mismatch going on there, even for a resident species. So if we think about Colorado, where I did my graduate research, this plot just kind of shows seasonality changing with elevation. So this yellow line that we have here, this is sort of a seasonality in prairie versus montane versus alpine. Well, we can basically see that if we measure this based on the number of growing degree days, in the alpine system, things are really constrained. So basically the growing season is much shorter, so the opportunities to breed are going to be much more limited. So we think that maybe in this type of system, that phenological mismatch could become a bigger issue. So we had two objectives for this. So the first objective was to develop mismatch indices, kind of like that figure that I showed from Miller-Rushing. So based on within season changes in vegetation and arthropod abundance. So that's what ptarmigan are eating whenever they're chicks. And then we also wanted to estimate relationships between the indices and chick survival to test for fitness consequences of missed time reproductive events, and doing this kind of at that individual level. So in terms of methodology, what we did is we simultaneously tracked temporal survival of ptarmigan chicks from the date of hatch to the date of fledgling age. And then we also tracked changes in resource abundance over that time period. So we tracked changes of plant and insect resources. And then we developed indices of phenological asynchrony. And then we fit those indices in capture-recapture survival models to check if chick survival basically varied as a function of mismatch. We evaluated these different models with Akayuki's information criteria. So we had different models and we wanted to see which ones were received the most statistical support. Yeah, so one really great thing about having a long-term data set is that we basically knew the exact places where hens would bring their broods after they hatched, because we had basically this 50+ year data set of locations of where those areas occurred. So we were able to use that information to delineate polygons around the brood location or the brood rearing areas, which also tended to encompass most of the nest habitat as well. And then we took those polygons and we generated random sample points where we actually measured changes in resources. So we repeatedly went back to these locations throughout the season, from the start of the season to the very end, to measure different types of resources. So we measured things like the forage species that were in bloom, if those were available or not, and changes in that. And we also measured NDVI, that's like a measurement of plant productivity, overall plant productivity. And we also developed insect transects, so we were able to develop changes in arthropod abundance over that time period as well. So at those points that I showed you, we had these locations where we took photographs with cameras that were specially modified to be able to take NDVI photographs. So these are basically these fixed quadrant frames of 1 meter squared. We could take a photograph of those every week, and then we could process those in some image software to calculate the NDVI values, the average NDVI values for those. So this is an unprocessed series of photographs from one plot. And then whenever we processed it with the NDVI software, you get something that looks like this. You can kind of see what's going on there at the very beginning of the season. There's not much productivity. It greens up and then it starts to senesce towards the end of the season. So what we were able to do with that information is develop phenology curves for resources. So the important one here is going to be the dark green ones in the background. These are our NDVI measurements that I just showed you with those that were taken with those cameras. So we can develop these curves, and that's great because now we can take the information from these phenological curves and extract information on the individual level covariates. So notice this is a 3x3 basically plot right here. So we've got for every year from 2013 to 2015 at every site we did this. So we've got site level information that we can connect then to the fitness measurements in terms of survival for the chicks. So whenever we do the modeling and we fit that covariate information to our survival models, we found that basically the model that had the best support included the mismatch index for NDVI. So whenever there's more asynchrony between when a nest hatches and the amount of resources available to it, there's a fitness consequence there, and that interacts with age. So the consequence is more severe for chicks that are at younger ages compared to those that are older. So we basically saw, across the board here from our model results that we've got this relationship, and then it seemed to be the most extreme at the Mesa Seco site, located in southern Colorado. So I think it's important to note for this, just to kind of rehash here. So we saw that the reproductive or the fecundity measurements that we recorded or observed in the field from that 1966 period up to basically the present, it was kind of declining, and we wanted to evaluate some potential reasons for why that might be, because we did know, we know that the phenology was changing, birds were shifting to nesting earlier. But I have to mention, for the work that I just showed with these, the effect on survival, that was limited to a 2013 to 2015 period. So we didn't have that detailed information to go back in time to specifically tie what we were seeing in that long-term data set to phenological mismatch. So it's important to keep that in mind. And we were also looking at the individual level, the level of the individual chick, to see if it suffered in terms of its fitness as a result of being, hatched at a time where it was asynchronous with its resources. But we did do some more recent work where we actually looked at more of like population level measurements using the observed survey data that we take in August, and we had that across all the years. And then from right around, I think 2002 or so, we start to get satellite imagery where we can calculate NDVI at a finer scale available to us in the form of MODIS imagery. So what we did in this plot right here, we're regressing the mean number of chicks per hen. So this is a kind of a population level metric against the mismatch index that we derived from the satellite imagery in MODIS. And we had, I think, about 15 or 16 points that we were able to do the regression on for this. And we see that this pattern does generally hold at the population level as well. So there's some good news to mention about this, and that's that if we look at that median hatch date and we regress that against the start of plant growth at our study area, we do see that it seems that the median hatch date tends to track that pretty well. So it seems, at that populations level, we still do have the ability for birds to be kind of flexible and show some plasticity in their behavioral responses or their breeding responses to keep pace with the resources, at least over the years that we've monitored them so far. So some future work on this, we have graduate students that are recently started up at Colorado State University with Cam Aldridge, who was my graduate advisor. And they're going to be looking at questions that are more closely related to genomics, or at least one of the students will be working on genomics, and asking questions about evolvability and, can birds basically kind of micro-evolve these traits? Is there enough genetic variation for them to respond to selection pressures to kind of keep pace with changing resource phenology? Okay, so that was my graduate work. I'm going to switch over now to a more applied example of some work that I did as a postdoc whenever I was out in California working with sage grouse. So the greater sage grouse is a declining sagebrush obligate species in Western North America. They have a polygynous mating system where males congregate on open arenas, which we call leks, and they display there in hopes of attracting females. So since about the 1950s, sage grouse lek monitoring has been the tool of choice by state agencies to monitor fluctuations in the abundance of their sage grouse populations. So lek data is like really key for sage grouse, and it's actually key for most lekking species of grouse. That tends to be like one of the most obvious types of data that you can collect in the field to get information about abundance and changes in abundance over time. So yeah, lek data, super important. So this was a recently published open file report that was put out by the US Geological Survey. And this used long-term lek count data to conduct a range-wide assessment of sage grouse trends. And it used data from all the occupied states in the US where sage grouse occur, And it did a really, really sophisticated series of analyses to look at overall trends by region, and all of that was completely dependent on LEC data. So LEC data gets a lot of use, but it also gets a lot of scrutiny too, since we have so much dependence on LEC data. And it's really our only large-scale source of information on changes in sage grouse abundance. So given that heavy reliance on that LEC data, and that management decisions that are commonly made are based on apparent trends in LEC counts, so there's an obvious interest in whether or not it's a reliable indicator of true population size. So the issue is that whenever we count birds on a LEC, we imperfectly observe the process, and we're not likely to see all of the individuals. So that has potential to introduce bias into any estimates that rely on it. So we can miss birds for a lot of different reasons, and these reasons include whether or not they're actually present. So typically the way that the LEC counts work is field biologists will go out to these LEC locations at a distance that's not going to disturb the birds in the LECs, and they use binoculars and spotting scopes, and they count the LECs, and they often will count the same LEC repeatedly multiple times in a season. And the goal there is to get to take the highest count, because the highest count should be the least biased, because you've got it's the highest count, so it should be the one that's going to be the most accurate. But we also have problems that are related to closure. So whenever these biologists go out there and make these counts, is it possible that if they're counting multiple LECs in a day, could we have movements of birds between those LEC locations to where maybe they count a bird at LEC A, and then they go to LEC B to count, and then they the bird from A moved to B, so they also count that one. So there's some potential issues of closure there. And then there's a typical problem of detection probability. So let's say we have 50 birds on a LEC. How good are we at actually counting all 50 of those birds? Is there some potential bias there? Maybe we miss some of them. And then finally, there's also this question of, are the LECs that we know about, is that a representative sample from the population as a whole? And because you can imagine, we're probably only counting a proportion of LECs. There are probably unknown LECs out there as well. So these are all important questions to consider. I just focused on the first two for this one project that I worked on out in California, and that was the problem of LEC attendance and closure. So the reason we were able to address this is because we had some fairly newly used devices that are placed on birds. They're GPS transmitters, and that allows us to kind of collect fine scale data. But our specific objectives for this were to estimate daily attendance probabilities within season. So those are LEC attendance probabilities, the probability that a bird that's in the population is going to actually be at a LEC on the day that we observe it to take our count. And we wanted to get those probability estimates both within season. So from LECing probably begins around March all the way through like May. Is there variation in that, or are those probabilities the same across it? Does it change by year? And then also if it changes among age classes. So we can usually identify sage grouse to a few age classes. So for example, subadult males and adult males, if there are some changes in LEC attendance rates among those, that has some implications for bias as well. So we also wanted to estimate that daily probability of switching between LECs within a season. And then again, we wanted to do that among the years also, and also among the age classes. So those were our goals. So the way we did this, our field sites were located in throughout Nevada. So we captured males in the fall and spring using spotlighting techniques, and that was done from 2013 to 2017. And then we marked males with 35-gram rump-mounted GPS transmitters. So we have a total of 67 males that we used in the analysis, and we recorded 1 to 3 morning locations with those devices, 1 hour before sunrise up to 2 hours after sunrise. And then for this to actually work, we also had to have LEC boundaries delineated. So to use the location data, we needed to know if those birds were actually going to be in, at a LEC or not, because this is all being done remotely. So we actually had to go out into the field and make sure that we had the boundaries properly delineated to where we could basically produce polygons in a GIS and then do intersections with the points and know if they were there or not. Okay, so this is maybe the part that might be a little bit confusing, so I'm going to try to go through this slowly. So on a given morning, a male sage grouse could basically be assigned to one of three possible location outcomes. So the first possibility, if a bird's location was off lek during the morning, it was recorded as a 0. So its outcome for that day was 0. It wasn't on a lek. The second possibility was that if a bird's location was on a lek, and it was also on the same lek as it was last observed on before. So in other words, it didn't switch leks. It was on a lek, and that lek that it was on was the same one that was previously observed on. The outcome was recorded as a 1, so it was on the same lek. And then the third possible outcome was if a bird's location was on a lek, but the lek location was different from the last lek it was on. The outcome was then recorded as a 2, so it actually switched leks, it changed. So finally, if we had a male that had 2 different outcomes recorded on the same morning, so for example, if he was recorded as being both on or off a lek, or if he was, what would be another scenario? If he was on the same lek followed by a different lek, so in either of those 2 cases, we would take the highest outcome for assigning it for that morning. Okay, so we had that information, so it was basically this 3 possible outcomes, and we used generalized additive mixed effects models with a multinomial link function because it wasn't binomial. It wasn't just a 0 or a 1. There were actually 3 possible outcomes for it. That's why we used the multinomial. And then we fit covariates for day of year. That was as a smooth term, so that allowed that probability to kind of like change non-linearly throughout the lekking season. We also included year as a fixed effect, age as a fixed effect, and then we had random effects fit to the bird and the site. So this shows what some of the lek delineations look like. So we had 77 leks among the 8 sites where we did the male captures. 19 of those leks were inactive, meaning that they hadn't recently had, in the past years of our study, there were never birds displaying on those leks, but 58 of those leks were active. And then this figure shows 3 leks at our Tuscarora site. That was a site that was located in northern Nevada. And the points are male locations recorded in the morning, and different colors represent different individual males. So to go ahead and just jump right to the findings from this. So this left-hand panel shows the daily probability of attending the same lek for adults and subadults. And there are basically 2 key points here to take away. So the adults attend leks at higher rates than subadults. So they're in that kind of blue shaded curve. And then the subadults peak much later in the season than the adults. So they attend at lower rates, and they also attend leks later in the season. So this right-hand plot shows the combined lek attendance curve. If we just pull that across adult males and subadult males, that's what that curve looks like. So I think the one clear takeaway from this is that there's a lot of variability within the lekking season in terms of lek attendance probability. So it does change, and that has implications because when you count the lek is gonna matter. Okay, so this next one shows the probability of switching between leks. So this left-hand panel shows that the daily probability of attending a different lek for adults and subadults, how that changes by day of year in the lekking season. And the key points from this are that the subadults are more likely to basically switch between leks compared to adults. And then if we combine both these together, so if we pull across the age classes, that blue plot on the right-hand side shows what that looks like. Okay, so we're curious about what the potential drivers might be for that annual variation in lek attendance probabilities. And I'm just now realizing I didn't actually show the annual variation in those lek curves. I just kind of showed the overall mean. But we found evidence that basically it does change by year a little bit as well. So some years the peak attendance probabilities might be higher, other years they might be a little bit lower. And we were curious like what was really driving that. So what we did is we took the mean peak attendance probabilities across sites for each year. And we didn't have a lot of data points to work with for this. So maybe take this with a grain of salt because we only have 5. 5 years. But we took those and we regressed those values against the cumulative winter precipitation that's on the top panel there. And then we also regressed those against the average winter temperature. And it looks like there might be a pattern there. So it appears to be that we have higher attendance or peak attendance probabilities whenever we have more precipitation. And we're not really sure why this is, but we think it does essentially have something to do with breeding propensity of females. Maybe whenever we have higher precipitation winters, that might mean more precipitation resources for early forbs and things like that. So females are more likely to breed. They're more likely to breed and be on leks. You know, we think that might affect lek attendance, bringing more birds onto the leks. Okay, and then finally, I've got this plot here that shows the effects of distance between leks on the probability of lek switching by year. So this is done for each year. So the probability of a male switching leks is much higher when the lek he was last on is in close proximity to another lek. So based on this relationship, you know, we see that there is some variation there across years. But it kind of seems like this probability of switching leks drops off considerably whenever we get around that 4 kilometer mark. So if there are birds that are on leks that are 4 kilometers or more away from other leks, their probability of switching between them is going to decrease pretty substantially. Okay, some general conclusions from this work. So the average annual daily peak attendance probability was 0.58, but it ranged from 0.33 to 0.83. Subadults attended the leks at lower probabilities, so 0.42 at peak, and they also attended later in the season. And then subadults were more likely to switch leks at 0.046 compared to adults at 0.019. So I mentioned, you know, earlier that these leks are often counted repeatedly within a lekking season. So biologists might go back to the lek 2 or 3 times and get counts, and then they take the maximum count. There have been some statistical modeling advances that actually use the information from those repeated counts to estimate detection probabilities. And if you can do that, you can actually correct the abundance estimate that comes out of those. So that's one potential that could be applied towards lek data to maybe kind of remove some of the potential bias due to these fluctuations in lek attendance. But I'd also say I think it's possible to overstate the problem of lek data as an index of abundance. I mean, I think we're pretty fortunate to have lekking species like grouse go out onto these leks and really expose themselves to where they're easy to observe and count. There aren't a lot of cryptic species that have that type of behavior. And there's still no question that, you know, lek data by itself is going to still provides information on abundance and changes in abundance. The question is really, you know, do we have some assumptions in the lek data itself that maybe would cause us to want to reevaluate if we're using that type of data for modeling, like what are the potential things that we could do to reduce biases that it might introduce, and just trying to get a better understanding overall of what that bias might actually look like. But I think there are, you know, new statistical approaches too that can really help mitigate a lot of this. Okay, so that's kind of ends this postdoc work that I did at the USGS. There are a couple other projects I worked on there too, but this was probably the one that I spent the most time on. I'm going to end here just kind of talking about some current work that I've been doing. So I've been working mostly with large-scale modeling of sage-grouse data sets. So that does include range-wide lek data across the Western US. And then we're kind of using that mostly in support of BLM projects. So a lot of habitat management types of projects are, we're applying these data sets towards those to figure out what conditions are associated with highest lek counts or lek persistence probabilities and those types of metrics. And then essentially building our statistical models and creating maps. We don't have any of these, so I've only been at this position, I guess it's probably getting close to 2 years, but I don't have any of those results to show because they're still in the peer review process, but that's what I've been working on for sage grouse. And then one of the other big projects I'm working on right now is creating range-wide seasonal habitat models for sage grouse. So this is a really, really big collaborative project. It's got over 70 collaborators that are a part of this. So they're basically contributing their VHF telemetry data sets, location data sets, to this process so we can build species distribution models or resource selection function models, and then get really, really fine scale predictions of used habitats across the occupied sage grouse range. So that's what I've been working on, and I'll probably continue to be working on for the next 2 years or so until these projects are complete. Another component of that work also includes doing some habitat forecasting as well, since we do have this temporal component in these telemetry data sets with years that have different timestamps. Okay, and then the ptarmigan in Colorado, we're still working on this, but I've kind of not as directly or heavily involved anymore. We actually have 2 PhD students that recently came to this project, and they're going to be doing demographic modeling and some genomics work. So they have access to the long-term data, and then they're also going to be collecting some of their own data starting this year. And we're looking forward to a lot of the stuff that I think they're going to be producing to answer some really important questions in the context of things like changing climate. And I'm certainly very, very excited about that. So I just want to thank Cam Aldridge. She was my graduate advisor for this, and also at the various different positions that I've worked at. Pete Coates and Mark Rica, whenever I was working out in California on the sage grouse project. And then lots of field crew members went into collecting these data sets that we analyze. So I want to certainly acknowledge them as well as our major funding sources. So that's all that I have for this presentation. I want to thank the RM-CESU again for the opportunity to present at this seminar. And if there's time, I'm happy to take any questions. Thank you.
Thanks so much, Greg. We do have time for questions. Again, I will ask you to use the Q&A box. Should be located at the bottom of your screen to type in your questions. And while we give folks a minute or so to do that, Greg, I'm just interested, what was the decision on the listing the whitetail ptarmigan?
Yeah, that's a good question. So the species was petitioned for listing in 2010, and that received a positive 90-day finding. But ultimately, I think it was in 2020 the decision came out for the southern whitetail ptarmigan. That's the one subspecies that occurs in Colorado, Altipetans. That was determined not to be warranted. So that was not listed as threatened or endangered. However, the subspecies that occurs up in Washington State, the rainieriensis, so that's around Mount Rainier, that one I believe is going to be listed as threatened. I don't know that that's official yet, but I know the Fish and Wildlife Service determined that that was like the proper, I think, listing for that species. So that's probably going to happen in the next year or two.
Great, we do have a question here. Let's see. Greg, given your findings on temporal variation and probability of attending LEX, what would you recommend for designing the surveys appropriate for the dynamic in-mixture model? That is, what is the temporal scale of closure within which the replicated surveys need to occur?
Yeah, I think if you use the in-mixture model approach on this, and I'll admit I'm not really an expert with in-mixture models, but I've worked with Adrian Monroe at Fort Collins Science Center, and he has a lot more expertise in these. But if you use the in-mixture model approach, that detection probability that is estimated is really going to be like this joint product of several distinct separate probabilities. So it's going to be like the probability of attending a LEC, the probability of observing a bird given, conditional on it being on that LEC. So it's going to be several separate ones. It's going to kind of estimate all those things jointly. So I think you can probably do that, apply that type of repeated count safely over that same window that is typically done for sage grouse, which is usually from sometime in like late March to late April. But you know, I don't know if Adrian would have different, Adrian Monroe would have different recommendations for that. But I think it's certainly something that should be considered in terms of going to that kind of repeated count type of design for counting LECs, because there does, you know, there is pretty clearly variation in the attendance probability. I should also point out that, you know, sage-grouse biologists have known this for a long time. They're not unaware of this. So that's one of the reasons that they did go with the max count approach. So if you go back to a LEC, you know, 3 times and you see variation in those counts, you want to take the maximum, right? Because that's going to be probably closest to what, you know, the true population number that's out there. But it's still not really a corrected by a detection probability type of estimate. So that would be my advice. I think maybe getting those repeat counts would be important if you're just going to stick with the LEC data and not go with like some type of other design. But there is also discussion too in the sage-grouse world about other ways that, you know, more systematic types of survey designs could be implemented that don't rely on LEC counts.
Are there any other additional questions for Greg? Well, while you might be typing something in, I'll just remind you that today's seminar is being recorded, and we'll have it available up on our website by early next week. And we'll send an email out with the link to Greg's seminar. I'll also point out that our last seminar in the series will take place on April 14th at 2 Mountain Time. And we will be joined by Michelle Frank. Michelle was our 2012 award recipient. At the time, she was working on her MA in history at Colorado State University, and she is now an assistant professor of US history at Utah State Eastern. And she will be presenting on toxic legacies, environmental cleanup in US West mining towns, and we hope that you will join us for that. I don't see any additional questions, so again, thank you all for joining us today, and special thanks for Greg for coming back, presenting your work that you received the award on, and also kind of getting us up to date of what you've been working on since then. So great stuff. Thank you so much, and we'll end it here.
Thanks so much, appreciate it.